Hello guys, welcome and welcome back to my channel. This is Dr. Prasanjit Das, academic JR in the Department of Pharmacology, AIMS Kalyani. And today I am here with my next seminar that is on the topic the gastrointestinal microbiome and the drug response. So, coming to the learning objectives. So, in this presentation, I will be discussing about the human microbiome, the pharmacomicrobiomics, then the direct and indirect drug metabolism, which is caused by the gut microbes, and the microbiome, which is affecting the pharmacodynamics of the drugs. That is what the drug does to the body, and finally, future of the drugs and the microbiome and their interaction. So, these will be the topics under which we are going to discuss we are going to discuss the microbiome and the drug response. Coming to the first slide, that is the human microbiome. So what do we mean by the word microbiome? So microbiome is basically a genomic collection of ecosystem of microbes. So these microbes include fungi, then we have bacteria, then we have viruses and several archaea. So all these together and their genomic material, that is the DNA, RNA, together comprises microbiome, and these microbes are usually present at a particular site in the human. So this is what is called as microbiome. And then what is the difference between microbiome and microbiota? So microbiota is nothing but the collection of the microbes, which I have already discussed, but they will not include the genetic material. Okay, so there is no genetic material involved in microbiota, it is only the collection of the microbes. Whereas in case of microbiome, there is genetic material also of the microbes. And this human microbe, that is the microbiome, is individualized for different body areas, and they have a greater diversity in case of the gut and oral cavity. So in oral cavity and gut, we have the greater diversity of the microbiota and the microbiome. Now coming to the composition of microbiome and the number of cells. The number of cells of the microbes is usually equal to the number of cells of the human at a particular body site. But the genes from the microbiome is totally high, and increased number of genes are seen as compared to the human genes at a particular site. Now coming to the members of the microbiota. So 90% of the gut microbiota are the members of two major bacterial phyla, that is Bacteroidetes and Firmicutes. And how this microbiome is established in the body. So the establishment of the microbiome begins at the delivery of the human being. So initially they will get the microbes from the mother and also from the environment, and depending on the mode of delivery, it will be depending on the mode of delivery that the number and the variety of the microbes will be different. So basically in cesarean section we will have different types of microbes, and in case of normal vaginal delivery there will be different types of microbes. And once the child is weaned, then the particular human being will get a signature microbiome, and it will be usually present throughout the life. But it can change following treatment with several drugs like antibiotics. Now continuing with the human microbiome, here you can see the microbiome, it is having a bidirectional relationship with the drugs, that is drugs will act on microbiome and microbiome will also act on drugs, and it is also having a bidirectional relationship with the diet. So diet also influences the microbiome and microbiome also influences the drug, and this microbiome is responsible for causation of several diseases, and it is also implicated in several diseases. So the interaction between the drug and the microbiome and the science which investigates this interaction is what we call as microbiomics, which will be dealt in the later part of this presentation. Now coming to some of the examples. So interaction of the microbiome with the drugs. As you can see, that gut bacteria reduces sulfasalazine, which is a prodrug, and it is the drug which is used to treat inflammatory bowel disease. So this is a prodrug which will be acted upon by the gut bacteria, and then it will form the metabolites, that is sulfapyridine, and the active drug, that is 5-aminosalicylate mesalamine, which is responsible for management of the inflammatory bowel disease. So this is one example where the gut bacteria is acting on the drugs. So there is an influence of the microbiome on the drugs. And now we have several antibiotics and non-antibiotics which damages the gut microbiota. So this is an example where the drugs are acting on the microbiome. Now coming to the other examples. So in case of mice, we have seen in studies that Clostridium orbicindens, it makes des-aminotyrosin by a hydrolysis process from the des-aminotyrosin conjugated flavonoids. So this flavonoids is basically a compound which is present in the fruits and vegetables, and this bacteria which is present in mice is responsible for forming the des-aminotyrosin, and this des-aminotyrosin in turn protects the mice from Haemophilus influenza virus infection. That is because this des-aminotyrosin is promoting the type 1 interferon signaling. So in this case, what we have seen is that the microbiome is responsible for acting on the diet, that is diet of the mice here. Then a ketogenic diet decreases the bifidobacterium, which is a pro-inflammatory bacteria, because it stimulates Th17 cells. So the ketogenic diet will lead to the production of the ketone bodies, which will decrease the bifidobacterium. And since there is decrease in the bifidobacterium, so there will be decrease in inflammation, and as a result, this strategy can be used for the treatment of diseases where there is inflammation, that is inflammatory bowel disease. Also, this gut microbiome is implicated in several diseases like autoimmune conditions and autism, because the gut microbiome can leak out of the gut and it can lead to damage to several systems like neurological systems, leading to autoimmune condition, neurological defects and autism. Now continuing with the action of the antibiotics on the microbiota. So antibiotics, they can damage the gut microbiota. So here you can see the antibiotics are usually given for some infections in the neonatal period and in the infantile period, and this will have a permanent effect on the microbiome of that particular individual. Also, we have other drugs like proton pump inhibitors when they are used for long term. So then antibiotics and long-term use of proton pump inhibitors will damage the normal microflora of the gut, and as a result the pathogenic bacteria will increase. So this will lead to a condition which is called as pseudomembranous colitis. So pseudomembranous colitis is actually Clostridium difficile infection. So here you can see, because of the action of the drug, there is decrease in the normal microflora, and this has led to the causation of an infection which is known as pseudomembranous colitis caused by Clostridium difficile. So now we will move on to the next slide, that is the therapeutic interventions which we can do using the microbiome. First is fetal microbiota transplantation, and this is nowadays used for the treatment of Clostridium difficile colitis, that is pseudomembranous colitis, another name for pseudomembranous colitis. So in this, basically we take the fecal matter from the healthy donor, which is an individual, and we will transfer it to the unhealthy donor who is having the Clostridium difficile colitis infection, and along with the fecal matter there will be transfer of the microbiota also. So this will help in the management of pseudomembranous colitis. Then we are also doing research nowadays on several other methods of treatment of some diseases like inflammatory bowel diseases. Then we have autism and phenylketonuria with the use of prebiotics, that is, these are the fibers which are usually digested by the microbes. So it promotes the growth of microbes, that is the healthy microbes, that is healthy bacteria. Then we have probacteria, which is actually the healthy bacteria, that is these are actually live bacteria which is given to the individual in a particular amount so as to produce the therapeutic effect. Then we have the postbiotics. These are nothing but the metabolites which are produced by the good bacteria. So these metabolites are only directly given into the individual, and this also produces a therapeutic effect. Now coming to the techniques for studying microbiome. So earlier in the time period, microbiome was studied with the help of the 16S rRNA. Here you can see the rRNA complex. Okay, so this is the rRNA, this is mRNA. So with the help of rRNA, we were studying the microbiome, and it was giving us information till the level of genus. Okay, so we were not getting information about the species of the microbes which are present in a particular microbiome. Now with the invent of newer techniques, that is metagenomic sequencing, a type of which is shotgun metagenomics, we are able to get the information which is even species specific. So in metagenomic sequencing, what we do is we will take the genetic material, that is the DNA and the RNA, which is present in a particular sample, and then we will be isolating the genetic material, and it will be fragmented, and then it will be sequenced, and finally we will see the different functions of the genes. So in shotgun metagenomics, this fragmentation which occurs, it occurs in the form of the pellets of a shotgun, just like how the pellets of the shotgun scatter. So that is the reason why it is also known as shotgun metagenomics. And to get expression level information of the microbiome, we do studies called as metatranscriptomics, metaproteomics, and metabolomics. So metatranscriptomics is basically getting the information about the RNA specifically, and then in case of metaproteomics, we are getting the information about the proteins present in the microbiome and the different protein interactions. And in metabolomics, we are getting the information specifically about the metabolites which are produced by the microbiome during the life processes. So this was about how the methods are used for studying microbiome. And along with that, there is one more method, that is using notobiotic mouse models. So notobiotic mouse models are basically the mouse models in which there are no germs, that is germ free mice, or they will be having one particular microbes only. So in that way we can introduce several other microbes in the particular mice, and we can see the changes which occurs in the mice, and then with that we will be able to find out that which germ or microbe is responsible for causing the particular changes in the mice. So this is the shotgun metagenomics. As you can see here, first we collect the sample, then from that we extract the DNA, RNA, and all the genetic material, then it is being sequenced and assembled, and finally we can see the different function and pathways for which this particular gene is responsible. So this sequenced DNA is what we call as metagenome, and this is the procedure of shotgun metagenomics. Here you can see the images of two mice. So this one is the notobiotic mice, that is the mice which is germ free, or it is having one particular microbe, and on the other hand it is the wild type of mice, that is which is having all the different types of microbes, as you can see here. So this is just a pictorial representation of notobiotic mice. Now coming to the next thing, that is the pharmacomicrobiomics. So it is the science which investigates the interaction between the pharmaceuticals and the microbes. So microbes, basically microbiome. So as you can see in this image, when a drug is being taken orally, it has to go through several processes, that is it has to be absorbed from the intestine, then it enters into the portal vein, then it goes to the liver, there metabolism can occur, then in intestine also metabolism can occur by the CYP enzymes, then some of the drugs can be directly effluxed back into the lumen with the help of P glycoproteins. So these are the several areas where the gut bacteria or the microbes can affect the drugs, that is they can either affect the metabolism of the drugs directly, or they can affect the metabolism of the drugs indirectly by modifying the host metabolic processes. So they can affect the phase 1 phase 2 reaction of the metabolism of drugs, or they can themselves directly metabolize the drugs. So the direct drug metabolism by the gut microbes. So you can see that bacterial metabolism of the drugs, it often results in increased hydrophobicity of the compounds, which is just the opposite seen in case of metabolism by the human microsomal enzymes, that is they make the drug more hydrophilic. As a result, that promotes the excretion. But hydrophobic compounds means it is more lipid soluble, and as a result there will be increased absorption. So this will help the absorption of the drugs back into the systemic circulation. So the different direct metabolism which the gut microbes does includes reduction, then we have acetylation, then we have methylation, then we have hydrolysis, out of which reduction and hydrolysis are the one to predominate. So hydrolysis, it is supporting the growth of the microbes, and reduction is supporting the anaerobic respiration of the microbes. As a result, these two are the reactions which are more predominant and occurs more. Now in this table you can see the example of the direct metabolism of the drugs. So reduction, under this we have several categories like azo reduction, then we have nitro reduction and reduction. So azo reduction means the reduction of the azo bond which is present, and then nitro reduction means reduction of the nitro group. So this is seen in case of the drug, that is sulfasalazine, which is an anti-inflammatory drug. So the different microbes which are implicated here are bacteroids, fragilis, then we have Streptococcus faecium, then we have Streptococcus faecalis. And after the metabolism, that is after the azo reduction of the sulfasalazine, it forms the active compound, that is 5-aminosalicylic acid. So as a result, it activates the prodrug, that is sulfasalazine. Then in case of chloramphenicol, it deactivates the drug. Then in case of digoxin, the microbe, that is Agarthala lenta, is responsible for deactivating digoxin. So what it does is it converts the digoxin into dihydrodigoxin, that is the digoxin's alpha beta unsaturated ring, it is being reduced, and as a result it leads to the formation of dihydrodigoxin. So it deactivates the digoxin. So the deactivation of the digoxin is by the activation of enzyme that is called as CGR, that is cardiac glycoside reductase 1 and 2. So if the person is taking a diet which is rich in arginine, okay, which is arginine, then it will reduce the activity of CGR1 and 2, and as a result there will be more action of digoxin because it is not converted into the inactive metabolite, that is dihydrodigoxin. Now coming to the other examples, hydrolysis, under which we have several categories, that is ester hydrolysis in case of a hypolipidemic drug, that is lovastatin, then deglucuronidation in case of irinotecan, which is an anti-neoplastic drug, so it is responsible for converting it into the active form, that is SN38G, that is activated. So the bacteria which is implicated here includes E. coli, then we have Lactobacillus rhamnosus, then we have Ruminococcus nevus, and Faecalibacterium prausnitzii. Then we have other examples like desulfation for sodium picosulfate, which is a laxative. Then we have dephosphorylation for 5-fluorouracil. Then we have amide hydrolysis for methotrexate, which is an anti-metabolite. Then we have deacetylation for diltiazem, which is a calcium channel blocker used as an antihypertensive. So these are the different examples. Now coming to the other examples, that is acylation. Under this we have propionylation and acetylation, that is addition of the propional group and addition of the acetyl group. It is seen in case of tobramycin, it is seen in case of aminoglycoside like kanamycin A. So in kanamycin A, it deactivates the drug and it is carried out by Mycobacterium tuberculosis. Now coming to the demethylation and decarboxylation. So demethylation, it is seen in case of the drug that is L-tretamine, which is an anti-neoplastic drug, and decarboxylation is seen in case of levodopa, which is used in Parkinsonism. So this deactivates the levodopa. Now coming to the indirect drug metabolism by the gut microbes. So as you can see, the intestinal bacteria, they can also indirectly affect the drug biotransformation, that is the drug metabolism, that is by regulating the host drug metabolizing genes. So in one study it was found that the mice, that is the notobiotic mice, which is germ free, was having decreased expression of CYP3A. So when the exogenous bacteria was given to this mice, that is conventionalization of the mice was being done, then it led to the formation of several microbes within the mice, and then it led to increased CYP3A expression in that particular mice. So as a result, there was increased metabolism of the drugs in these mice. So this proves that the microbes can alter the drug metabolism indirectly also. Now coming to the example of an analgesic, that is acetaminophen. So here the gut microbes alter the phase 2 metabolism of these drugs. So the drug acetaminophen is metabolized by the reaction, that is glucuronidation, which is the phase 2 metabolism of the drug. So after this, the glucuronide conjugate of acetaminophen reaches the gut, and there the bacteria present in the gut is having beta glucuronidase enzyme which deconjugates the acetaminophen. As a result, it is being absorbed back from the gut, so then it enters into the enterohepatic circulation. So if we use some antibiotics which decreases the number of the microbes which produces this beta glucuronidase enzyme, then it will lead to the decreased action of acetaminophen because it will prevent the deconjugation. And also there are some gut bacteria which produces a compound called as paracrisol that competes with acetaminophen for binding to sulfotransferase enzyme. So sulfotransferase enzyme is the another enzyme which actually acts on the deconjugated acetaminophen and metabolizes it further. So if there is decrease binding of acetaminophen to sulfotransferase enzyme, and then there will be increased production, so there will be increased levels of the active acetaminophen, and this will lead to hepatotoxic effects. So hepatotoxic effects are seen in case where there is high levels of paracrisol. This leads to the lower capacity to sulfonate the acetaminophen and leading to hepatotoxic effects. And there are several other drugs where there is indirect drug metabolism by the gut microbes. Examples include caffeine, metformin, omeprazole, etc., and so many more examples. Coming to the microbiome, that is affecting the pharmacodynamics, that is the drug doing action on the body. So we can see in case of abundance of Faecalibacterium, which is a bacteria in melanoma patients, they are responding to immune checkpoint inhibitors therapy better. That is, they are having a significantly longer progression free survival, whereas the patients who are having relatively higher abundance of another bacteria which is called as bacteroideals, they are having a shorter progression free survival with treatment of immune checkpoint inhibitors therapy. So these two bacteria, that is Faecalibacterium and bacteroideals, they are actually regulating the response of the drugs, that is immune checkpoint inhibitor, in the patients of melanoma. Then another example is Fusobacterium nucleatum. So Fusobacterium nucleatum is associated with shorter survival in colorectal cancer patients. And this Fusobacterium nucleatum is actually responsible for shorter survival in the colorectal cancer patients because it controls the chemoresistance of 5-fluorouracil, capecitabine, and oxaliplatin. These three drugs which are commonly used for the management of the colorectal cancer. So with these two examples, you can see the microbiome is also affecting the pharmacodynamics of the drugs. Now coming to the future of the drugs and the microbiome. So in this, first we will be discussing about the fecal transplant. But before going into the fecal transplants, let's discuss what is called as transformation. So transformation is basically the transfer of the microbes from a healthy livestock into a unhealthy livestock. So this is used for the management of diarrhea, then constipation, and decreased milk production in the livestock. So the same counterpart in human being is called as fecal transplant, which is also known as bacteriotherapy. So the donor feces, which is containing the good microbiota, is being mixed, blended, and filtration occurs, and finally then it is being introduced into the unhealthy individual, either in the form of oral capsules or via endoscopy. But the FDA has not provided approval to the use of this fecal transplant completely. They have provided a current guidance, that is enforcement discretion under limited conditions. So these conditions in which we can use the fecal transplant include: the licensed healthcare provider who is treating the patient should obtain an adequate consent from the patient or his or her legally authorized representative for the use of FMT products, that is fecal microbiota transplantation products, and the consent should include a minimum statement of that the use of FMD products to treat Clostridium difficile infection is investigational, and a discussion of the reasonable foreseeable risk should be mentioned. Also, the FMD products should not be obtained from a stool bank, and the stool donor and the stool are qualified by screening and testing. So these are the conditions which, when present, then only we can use the fecal transplant for the management of Clostridium difficile infection, that is pseudomembranous enterocolitis. Now continuing with that, so FMT, that is the fecal microbiota transplantation, is nowadays being evaluated for the management of several other diseases also, like Crohn's disease, that is inflammatory bowel disease, ulcerative colitis, which is also an inflammatory bowel disease, irritable bowel syndrome, pediatric cytomegalovirus disease, then autism and obesity. But there is no standardized control process for the preparation of this fecal transplant, and as a result there is increased chance of transmission of microbes including HIV, that is human immunodeficiency virus, and SARS-CoV virus via the fecal transplants from the healthy donor to the donor in which we are suspecting the particular disease, that is Clostridium difficile infection. And following a successful fecal transplantation, there can be several other side effects like diarrhea, then there can be loose stools, that is changes in the consistency of stools, then there can be bloating, there can be cramping, and several other side effects. So these are some of the disadvantages of using the fecal transplants, and that is why the FDA has also not approved fecal transplant from all the conditions. And in fecal transplants we are having several other problems also, like after the fecal transplant the recipient can actually get the metabolic characteristics of the donor. So there can be weight gain, there can be loss of weight also, and all the other side effects like constipation, diarrhea, cramping, bloating, I have already mentioned, as you can see here. So we are nowadays thinking of other methods, like because in fecal transplant we are getting a variation in the efficacy, so we are now trying to use single strain microbiota, which includes the probiotics like E. coli, Neisseria, then Bacillus subtilis, then Lactobacillus bacillus, or we can use the bacteriophage, as you can see, the viruses which are infecting the pathogenic bacteria and then killing them. So these are the methods which are being nowadays tried, and research is being going on in these methods so as to treat the pathogenic bacterial infections. Now coming to the next method, that is the designer microbiota. So these are the genetically engineered microbial strains. So they are used to deliver specific therapeutics, and they can also make up for several deficiencies in the body. One example which we can take is phenylketonuria, which is occurring because of the deficiency of the enzyme that is phenylalanine hydroxylase, or it occurs because of the deficiency of the cofactor that is tetrahydrobiopterin. So this can be managed by the use of several microbiota like E. coli and Lactococcus lactis, which are engineered to contain the enzyme phenylalanine hydroxylase, and as a result this will reduce the phenylalanine levels, so this will lead to the management of phenylketonuria. So this is one example where designer microbiota is use in some of the diseases, and several other diseases are being researched for which we can use the designer microbiota. Now coming to the phage therapy. So phage means the bacteriophage. So these are the viruses which infect the bacteria and and then finally they kill the selected bacteria. So phage therapy is being nowadays evaluated in several clinical trials as a strategy to treat drug resistant urinary tract infection. Then we have Klebsiella pneumonia infection. Then in case of Burkholderia cenocepacia infection, then Pseudomonas aeruginosa infection, Staphylococcus aureus infection, and the other multi-drug resistant pathogenic infections. And it is also used to treat inflammatory bowel disease, then lung infections, osteomyelitis, mastoiditis, prosthetic joint infections. Here you can see the bacteriophage which is affecting the toxic bacteria. Now finally coming to the take-home points. So the gut microbiome plays a crucial role in drug metabolism, and as a result it influences the toxicity and efficacy, both toxicity and efficacy of many medications. And modulating the gut microbiome through probiotics, prebiotics, or fecal microbiota transplantation may serve as a potential strategy to optimize the drug delivery and enhance the therapeutic outcomes which we are getting from the drugs. So these are the two major take-home points from this seminar, and these are the references which I have followed, mainly Goodman Gilman, the Pharmacological Basis of Therapeutics 14th edition. So with this I would end this seminar, and thank you guys. So I hope you like this video. If you like my content, please do like, share, subscribe, and also press the bell icon to get notified whenever I upload a new video. Thank you guys.